In this activity, we'll be examining Elodea, a common aquatic plant that is widely used in biology labs because its cells are easy to observe under a microscope. This activity will allow you to clearly see chloroplasts and understand how plant cells are structured to carry out photosynthesis. Elodea is an excellent model organism for studying plant cells because its leaves are extremely thin, only about 2 cell layers thick. This allows light to pass through easily, making it possible to observe individual cells and organelles without special staining. Because Elodea lives underwater, its cells are well adapted for efficient photosynthesis. Chloroplasts are specialized organelles responsible for photosynthesis. They contain chlorophyll, the green pigment that captures light energy. In Elodea cells, chloroplasts are often visible as small green bodies moving around the edges of the cell. As you observe, think about how their position in the cell supports their function. Before you begin observing, take a moment to think about this question. Why might chloroplasts in aquatic plants like Elodea be pushed toward the edges of the cell rather than staying in the center? Consider how light enters the leaf and where it would be most effective for chloroplasts to be positioned. Pause the video and write your prediction in your lab notebook before moving on. A single Elodea leaf is placed on a clean slide with a drop of water and a cover slip. As it is placed at scanning power, this is how it appears. Focus near the edge of the leaf where the cells are thinner and easier to see. At this stage, you're looking for the general shape and arrangement of the cells, not fine details yet. Now we've switched to high power, where you can start to see individual Elodea cells much more clearly. As you look at this image, start asking yourself, where is the boundary of each cell? Can you trace the cell wall all the way around? Do you see small green structures inside the cell? Where are they concentrated? Near the edges or throughout? Are the green structures all the same size and shape? Is there a large, clear, empty-looking space in the center? If so, what might that tell you about the vacuole? Do you notice any movement? Are the chloroplasts staying still or slowly shifting? The movement of the chloroplasts around the vacuole is called cytoplasmic streaming. Where is the cytoplasm located? Is it spread evenly or mostly along the edges of the cell? Can you find the nucleus? If you can't, what might make it difficult to see in this cell without staining? And just like with the onion slide, at this point, resist the urge to label everything right away. Slow down, observe carefully, and think about what each structure might be doing. Now let's take a closer look at what you're seeing in the Elodea cells. The cell wall forms the rigid outer boundary of each cell. It gives the cell its shape and provides structural support. Just inside the cell wall is the cytoplasm, which is the fluid portion of the cell where many cellular processes occur. In Elodea, the cytoplasm often appears as a thin layer lining the inside of the cell. You should notice many small green structures along the edges of the cell. These are the chloroplasts. Chloroplasts contain chlorophyll and are responsible for photosynthesis. In Elodea, they are often pushed toward the outer edges of the cell. The central vacuole takes up most of the interior space of the cell. You may not see a clear membrane around it, but you can infer its presence by the large clear area that pushes the cytoplasm and chloroplasts toward the cell wall. The nucleus may be difficult to see in these cells. If visible, it appears as a lighter, rounded structure within the cytoplasm. It can be hard to distinguish without staining, so don't worry if you're unable to clearly identify it. As you observe, focus on how the placement of each structure supports the cell's function, especially the role of chloroplasts in capturing light for photosynthesis. In your lab notebook, draw one Elodea cell that represents what you observed. Be sure to label the cell wall, cytoplasm, central vacuole, and chloroplasts clearly. Your drawing should be neat and proportional, but it does not need to be artistic. Go ahead and pause the video to complete your drawing. Now that you've observed the structure of Elodea cells, we're going to look at how water movement affects those cells. This process is called osmosis, and it plays a major role in maintaining the shape and function of plant cells. As water moves in and out of the cell, it changes the pressure inside the cell, which directly affects how the cell looks under the microscope. Osmosis occurs when water moves across a membrane from an area of higher water concentration to lower water concentration. In plant cells, this movement affects turgid pressure, the pressure of the cell contents pushing against the cell wall. This pressure is what keeps plants upright and firm. When a cell is placed in different solutions, water moves depending on the concentration of the solutes. Tonicity is the measure of a solution's ability to change a cell's volume by affecting water movement. In a hypotonic solution, the fluid outside of the cell has a lower solute concentration than the cell, so water enters the cell, making it firm or turgid. This is the normal, healthy state for most plant cells. In an isotonic solution, the fluid inside and outside the cell is the same, so water movement is balanced or flaccid, and the cell maintains its normal volume. In a hypertonic solution, the fluid outside of the cell has a higher solute concentration than the cell, so water leaves the cell, causing the cell membrane to shrink away from the cell wall. You may initially see plants wilt. After prolonged exposure, the cells can die. Before we begin the osmosis portion of the lab, take a moment to think about this question. Turgid pressure is created when water fills the central vacuole and pushes outward against the cell wall. This pressure plays an important role in the structure and function of plant cells. Based on what you already know about plant cells and their role in supporting the plant, why do you think maintaining this pressure is important? Write your prediction in your lab notebook. There is no single correct answer at this stage. What matters is your reasoning. As you move through the activity and observe changes in the cells, you'll come back to this question and refine your understanding. Go ahead and pause this video. As you observe Elodea under different conditions, watch for changes in where the chloroplasts are located. In a hypertonic solution, you may see the cytoplasm pull away from the cell wall, a process called plasmolysis. These visible changes help demonstrate how osmosis affects living cells. In this GIF, you see Elodea in a 20% sugar solution. So if you ever tried that science experiment where you water plants with soda, this may be what you see at a microscopic level. What would the plant look like with your bare eyes? This video clip is a few minutes after I added a couple of drops of 10% salt solution at the edge of the slide cover. You can see under the low power that the chloroplasts are bunched up in each cell as the central vacuole releases its water as a way to balance the solute concentration. As I move up to a higher power, this should be seen even more clearly. Now it's time to draw a plasmolyzed cell in your lab notebook. You can label it Elodea in 10% NaCl. Focus on the new position of the cytoplasm and chloroplast in relation to the cell wall. Pause the video so you can draw it. Think about the shape, chloroplast presence, and function of the onion and Elodea cells. Both are leaf tissue, but they look very different. Elodea has chloroplasts, while onion cells do not. Why is that? Think about what environment these parts live in and their purpose. Pause the video to answer this question. Think about the two Elodea cells. One is hypotonic and the other is hypertonic. What changes did you observe after the salt solution was added? Let's get more practical and apply this to the world of horticulture. Fertilizer, especially the quick-release fertilizers, act like a salt, and you may hear the phrase that too much fertilizer burns the plants because you often see leaves with a scorched appearance like fire hit it. As the leaf cells absorb the fertilizer solution, what is happening to the plant cells? Pause the video to answer these questions. The Elodea and osmosis activities work together to show how plant cell structure and water movement are closely connected. Elodea leaf cells are specialized for photosynthesis, which is why chloroplasts are abundant and positioned along the edges of the cell where light can be captured most efficiently. The large central vacuole plays a key role in maintaining turgid pressure, keeping the cells firm, and supporting the overall structure of the plant. When water moves into or out of the cell through osmosis, it directly affects the shape and function of the cell. These observations help explain real-world plant responses, such as wilting, when water is limited, or when plants are exposed to high salt conditions. Understanding these relationships will be important as we move into later labs on plant tissues, transport, and photosynthesis.